Hey guys, it's Ms. Gruman here, and I'm going to be talking to you guys about photosynthesis and cellular respiration lab, and we're going to be using algae beads to help us model this. Things that you guys need to complete prior to. So you guys need to take notes on this presentation. You need to look up which scenario you are performing. You need to download the lab report document and fill out and create the following. You need to look at the objective. I wrote most of it. It's just fill in the blank at this point. You need to create a null and alternative hypothesis. You need to identify which independent, dependent variables and controls if it's applicable to your scenario. And then I created a data table for you guys. You just need to specify the titles and adjust it so now it works for your specific scenario. So the objective today is that you guys are going to learn the background information in order to perform the laboratory experiment with us analyzing the connection between photosynthesis and cellular respiration. The four setups that we're going to be analyzing is that we're going to be looking at light intensity on metabolic process. Remember, the metabolic process could be either photosynthesis or cellular respiration or both. Light distance on the metabolic processes, color spectrum affecting the metabolic processes, and temperature affecting the metabolic process. So we're essentially going to be analyzing this diagram where we're linking the chloroplast and mitochondria. The reason that we're going to be using algae beads, which are located right here, is because these algae beads are going to be autotrophs. So we're going to be using autotrophs because they can do both and they have both of these organelles. So we're actually not going to be analyzing the organisms themselves. We're going to be analyzing the gases that they emit. So take a second and think about what are the two gases that are nonpolar that can be emitted in cellular respiration photosynthesis. So after thinking about it, you should come to the conclusion that oxygen could be emitted that we could analyze coming out of the environment, or CO2. So these are the two ways that laboratory experiments analyze the cellular respiration rate or photosynthesis is by either looking at CO2 levels or looking at oxygen levels. They just pick which gas they want to analyze, and that's what they do. For us, we're going to be analyzing CO2 gases and seeing how much CO2 gas is in this liquid. And from us analyzing that, we can see, is it cellular respiration that's on, or is it photosynthesis that's on? Which metabolic process is taking effect, and how well are they doing their job? So I want you guys to take a second to think about, when do autotrophs perform photosynthesis and when do they perform cellular respiration? Is it a simple on and off switch? Are they both running at the same time always? How does it work? So after thinking about it, this is a graph that really shows you how it works throughout the whole day. So we have our time down here that's showing like a 24-hour interval. And what we can see is that actually cellular respiration is on all of the time. It's a very low content of cellular steady state that cellular respiration is on. And that makes sense because we need ATP. ATP is used to fuel everything inside the cell, not just the mitochondria. So because of that, we should be synthesizing all the time. What happens, however, though, is that when there's sunlight or photons, is that photosynthesis is just cranked up, that there's more oxygen being emitted out than CO2 in comparison when I'm looking at the ratio. So that's why it appears that only photosynthesis is on during the day, but the actuality of it is actually that's not true, and that cellular respiration is at its highest point during the day. So also take a second to think about, why would cellular respiration be, even though it's just slightly higher during the day? Think about how enzymes perform and like optimum temperature and things like that, and what is provided during the day that can maybe increase those enzymatic functions. So how are we going to analyze cellular respiration photosynthesis? Simplistic answer, we're going to be looking at the pH scale or absorption if we were using a spectrophotometer. What you guys are going to be receiving are these cardboard cards, and you're going to be analyzing the liquid, like I said. So you're going to be looking at what color is this liquid, and that's going to correlate with pH, and it's also going to correlate with absorption rate. So remember, the more absorption that's being used, the higher that rate that light's being used, and lower the absorption, the less likely that light's being used. So again, Just to emphasize, we're going to be analyzing the liquid, not the actual algae beads themselves, and we're going to be looking at the color change of the liquid. And the way that we're going to get color change is by how much CO2 enters the liquid. So if CO2 is entering the liquid, that means it must be leaving the algae beads, and vice versa. If CO2 is leaving the liquid, then that means that CO2 must be going into the algae beads. So this is the chemistry behind the pH and how we can use it. So in a normal situation, we would mostly have water, but we do get CO2 that enters the environment. And when CO2 enters the environment, it creates this bicarbonate. So the more CO2, the more bicarbonate we create. And the more bicarbonate that we create, the more dissociation and free hydrogen ions that we have floating around in the solution. So the more hydrogen ions that we have in solution, think about it. Does that make it more acidic or does that make it more basic? So actually, the more hydrogen ions we have, the more basic it is. So there's a correlation that the more CO2 in the environment, the more acidic we'll be because we will have more hydrogen ions present. So again, we're going to be analyzing the liquid. So we're going to be looking at how much hydrogen ions are in the liquid. And the reason we would get more hydrogen ions is because we would get more CO2. So now just take a second and think about which process would allow more CO2 to end up in the liquid. Would it be the photosynthesis or would it be cellular respiration to have CO2 leave the algae beads and go into the liquid? And then vice versa, think about what scenario would allow CO2 to leave the liquid and go back into the algae beads? And how would this affect pH? So go ahead and create this cuvette diagram into your notebook, and I would even draw arrows, kind of like how I'm pointing with this little guidance thing, and draw like what would happen to the pH in the liquid if CO2 was going out, and what process would this be, and what would happen to the pH if liquid CO2 was going into the algae bead, and what would happen to pH. So now I'm going to go over to the 4 different types of versions that we're going to be doing in this lab. One is going to be on light intensity of the metabolic process. With that one, everybody's going to have a prep step that's going to be printed out for you guys to do the directions. It's less than 5 minutes to set up these algae beads. We just have to wash them and put them in fresh solution. But if you receive the light intensity, you're going to receive 4 different cuvettes, and the cuvettes are going to be colored differently. So one's going to have no color, so that's going to be that there's no filter on it. There's nothing blocking the light from entering it. One's going to be completely covered. That's going to be 100% blocked from light. We're going to have 50% shaded so that there's going to be dashes on the cuvette. And the last one is that there's going to be dashes in 2 directions, and that'll be 85% shaded. What I want everybody to do, whether you're assigned this group or not, is to answer the questions. Do we have a negative or positive control in this scenario? If we do, which one would represent that? Which process would be on at each filter paper? I don't want to say on. Which one would be most dominantly present? Would it be photosynthesis or cellular respiration? Based on that, what do you think would happen to the acid and base level? Would you see an increase or decrease in this in comparison to everybody else? So again, you're going to be answering the questions whether you're given this scenario or not. So one is going to be light distance. So light distance is very similar to light intensity, but instead, this is going to be 8 cuvettes. So this is going to be 2 groups working together. So with this one, what you guys are going to be doing is that you guys are going to be given one source of light. One group is going to receive 4 cuvettes with no filter on it, so there's going to be no coloration on it, and they're going to be setting those at 4 different distances and measuring the rate that they would see and see what would be on and what is the rate of that metabolic process the further I get from the light. And then we're going to be doing the same thing, but instead we're going to be blocking the cuvettes. So now no light can penetrate it, but we're still going to put it at various distances and see, is anything affected by the metabolic process, whether they can have access to light even if they're blocked or not blocked. So go ahead and answer these questions. That's posted, so you can go ahead and pause it. So the next one is that we're going to be looking at color spectrum. So now instead of like a black and gray filter, we're going to have 4 different colors of filter paper. So again, instead of doing distance, all the cuvettes are going to be at the same distance, but they're just going to have a color filter on top of them. So you have to think which color would be more predominant for photosynthesis and which color would be the least dominant. So if it's the least dominant, does that mean cellular respiration would be on? And how do you think that would affect the pH scale? And the last one that we're going to be looking at is temperature. So I'm going to be setting up 4 beakers for you. One's going to have boiling water in it, one's going to have ice water, one's going to be room temperature water, and the other one's going to be warm water. So this one, we're going to be having 4 that are completely color blocked, and then 4 that are not blocked at all. And we're going to be evaluating their metabolic rate based on that. So we have to think about, does cellular respiration do better in cold or warm or hot? And same thing with photosynthesis. So really consider enzymes in this scenario and how enzymes are affected by varying temperature. So that's it for this presentation. I hope you found it helpful, and I can't wait to do this lab with you guys.